So we're right back at it again, talking about this Chapter 8 material from the textbook. This is the second half of the Lecture 8 series on lipids and biological membranes. We've already talked about lipids in brief, spent a good amount of time on that in the last chunk, and with that in our back pocket, we're almost poised to move on to biological membranes and talk about their general biochemical characteristics and shapes and structures. From there, we'll talk about membrane proteins in general, classify the different types of membrane proteins, and get at this fluid mosaic model that I keep referring to as a way of thinking about how these lipids and these proteins work and move together in a standard biological membrane. We'll end up talking about active and facilitated transport and the roles of pumps and channels used to get molecules across the lipid bilayer. But before we start talking about bilayers and membranes in general, we've got one more class of lipids that we need to discuss that we really didn't have time for in the last chunk, and those are the cholesterol-based molecules, the steroids. As we said in the last chunk, steroids all share a general similar structure, this fused ring idea. 4 rings that are fused together and very, very hydrophobic. Lots of hydrocarbons coming off the ring here in cholesterol at least. And really the only thing that cholesterol has that is even remotely polar is the single hydroxyl group. And this general structure of cholesterol can serve as the backbone for creating a wide variety of different molecules that have a wide variety of functions. Some of the hormones that dictate our endocrine system and allow our endocrine system to work are based on a cholesterol backbone. These are the steroid hormones. And examples are the male sex hormone testosterone and the female sex hormones estradiol or estrogen and progesterone. All of these are based on cholesterol. All of them are steroids and all have this fixed or fused ring structure. You can see that testosterone, estradiol, and progesterone are all more hydrophilic than their cholesterol precursor because they have some polar bonds here, some partial charges up here. Even this carbonyl carbon is slightly polar, but all of them largely hydrophobic. Cholesterol itself is also considered a steroid. All of it falls into the same category. Perhaps more importantly than being the precursor for steroid hormones, though, cholesterol is actually an important and integral part of lipid bilayers in eukaryotic cells in general. As we've already noted, the only polar thing about cholesterol is that single hydroxyl group, but still that is enough to get cholesterol to engage happily in the lipid bilayer. The rest of cholesterol is highly hydrophobic, and so cholesterol is able to slip into the eukaryotic membrane and actually stiffen it up. That's how cholesterol causes heart disease, in fact, is by causing the membranes of the cells that make up the arteries to stiffen, become less pliable and less flexible, and that leads to heart disease. But with bad always comes good, because without cholesterol we wouldn't be able to make our hormones, cholesterol serving as the precursor for that, and sometimes cells need to make a firmer or denser membrane, and cholesterol plays a role in that as well. So we'll come back to cholesterol as we talk about lipid bilayers a little bit more here in the future, but we've got all the ingredients now, all of the parts we need to begin talking about biological membranes from the biochemical perspective. So there are a few things that are shared among all living cells on this planet. One of those things is DNA, and one of the only other things, is the presence of a plasma membrane. Every single living cell on this planet has a plasma membrane that's largely structured the way we're about to discuss. Eukaryotic cells are different and unique in that they also have membrane-bound organelles, but those organelles are a later evolutionary addition to what cells already had, which was the lipid bilayer itself. Cellular membranes are made up of both lipids and proteins, as you've already surmised, I'm sure. So we'll talk about the lipid component of membranes first, and then we can introduce the different kinds of proteins that exist. It's important to note that each are equally important for a membrane doing the job that it needs to do. The lipids and the proteins are equally important. And membranes serve 2 primary jobs. One is to protect the cell from that dangerous entropic world that we talked about in the previous lecture. And the other, at least for eukaryotic cells, is to allow the formation of these compartments, to allow cells to specialize individual areas for individual functions. So membranes allow cells to be more robust and more complicated, while also protecting it from the harsh world that's out there. If you dig a little deeper and you talk about membranes being a barrier or boundary or protective layer from the outside world, Well, then you can kind of think about membranes as gatekeepers in a way. Some of the things in that big bad outside world we need inside the cell, like glucose and oxygen. Other things from that big bad world we never want in the cell, like toxins and mutagens. And so membranes then become critically important in regulating what enters the cell and what does not enter the cell, what can leave the cell and what can't leave the cell. So membranes are the cellular gatekeepers of our cell. Again, we have membrane-bound proteins. These membrane-bound proteins can be enzymes. They can be receptor proteins, receptors that pay attention to and pick up signals from the outside world and transmit those signals inside the cell. And we have these transport proteins as well, such as pumps and channels that we have alluded to already and we'll talk about later on in the lecture. All of these membrane-bound proteins exist for the benefit of the cell. Each one of them has a dedicated job and function, and all of them require the membrane to do their job properly, truly making them membrane-bound proteins. The principal component of all cellular membranes are, as we've already said, these phosphoracylglycerols or phosphoglycerides, these amphipathic molecules that have two fatty acids and a very hydrophilic head group, comprised of a phosphatidic acid and alcohol as well. And these phosphoglycerides make up the lipid bilayer. And here they are, we can see them. These are the schematic representations of the molecules we talked about in chunk A. This blue circle is meant to represent the very hydrophilic alcohol group and phosphatidic acid. It also includes the glycerol. These squigglies are meant to encompass the fatty acid tails, the two fatty acid tails that we see in these phosphoglycerides, and only two because that third slot is being held by the phosphatidic acid. So these phosphoglycerides make up the lipid bilayer, and as we said at the end of the last chunk, they're kind of opposed to one another. They stand together feet to feet, where the fatty acid tails that are nonpolar and hydrophobic are huddled together inside the membrane, in the interior of the membrane, shielding them from water, and the hydrophilic groups that are more capable of interacting with water are on both the inner and outer surface of the membrane, interacting with the watery environment that we encounter inside and outside the cell. So, largely cells are bags of water. Of course, the extracellular environment is very water-based as well, and those hydrophilic groups interact favorably with all of those watery environments. The proteins, although they're not shown here, can make up anywhere between 20% of a membrane, all the way up to 80% of a cell's membrane for specialized cells that require a lot of membrane-bound proteins. And so it's not as though every once in a while there's a protein. Proteins are a very large component of our cellular membranes. And these membranes also contain the occasional glycolipid, which we also defined in the last chunk. These are lipid molecules that are fused to sugars, important for signaling, among other things. And they also contain steroids, most commonly cholesterol, to thicken their membrane. But there's a cholesterol-like molecule that we find in plants called phytosterols, and they do the same job in plants. They stiffen the membrane. So if you think about it, almost frighteningly, the only thing holding this boundary, this border, this protective layer around our cells is the mutual dislike of water that the hydrophobic tails have. That's the driving force for the membrane assuming the shape that it has. Now, once the presence of water causes those hydrophobic tails to huddle close together, then we can get induced van der Waals interactions because those molecules, those atoms, are at van der Waal distances. But really, it's nothing more than hydrophobic forces that are causing these membranes to form. Additionally, those hydrophobic forces also allow lipid bilayers to have a property that's very, very useful. We call that property self-sealing. So these membranes are self-sealing. Imagine that this is a piece of a cell membrane that we just manipulated. Imagine we sheared that cell membrane in half. We've created this huge hole in the membrane that we see here, all within the context of the larger cellular environment. What are these hydrophobic tails now exposed to that they weren't exposed to a moment ago? Water. And they do not want to be near that water. In fact, they will do anything and everything they can to shield themselves from water by huddling with other hydrophobic things. And the nearest other hydrophobic things are the other hydrophobic tails nearby. So this entire hole self-seals. so that we have a continuous layer of hydrophilic heads on the inside and a continuous layer of hydrophobic heads on the outside, and all of the tails then return to the interior of the membrane, restoring it to normal. So these membranes self-seal, and that self-sealing is driven by nothing more than trying to shield those tails from water. So, surprisingly, or perhaps not surprisingly, based on the name, the lipid bilayer is made up of two layers, but more importantly, those two layers are different. The outer and inner layers are distinct and unique from one another. One way that these two layers are different from one another is in the asymmetry of the polar head groups. The polar head groups on the outer surface of the membrane that we see here tend to be much larger and bulkier than the polar head groups inside, with the interior face of the membrane having smaller head groups and the exterior face of the membrane having larger head groups. We introduce this natural curvature to the cell membrane, which allows the cell membrane to take on that spherical shape. You can imagine if these head groups are large and bulky, and these head groups are small and compact, the inner face or the inner surface of the membrane takes up less space, while the outer surface takes up more, and we get this natural bend or curve to the cell membrane, giving the cell its spherical shape. Additionally, if the tails, those hydrocarbon tails of the bilayer, are ordered and dense, if they can pack close together, we have a more rigid membrane. If the tails of the bilayer are disordered and loose, the membrane will be more fluid. In bacterial prokaryotic cells, cells control the rigidity of their membrane by controlling how many double bonds are present in those fatty acid tails. The more saturated the fatty acid tails, the denser those fatty acids can be, the more rigid the membrane will become. The more double bonds you introduce, the more unsaturated those tails are, the less rigid they will be, the more fluid the membrane will be, and therefore we have a less dense membrane. That's how bacteria do it. We don't do it that way. For us, the fluidity of our membrane depends on what's present in that membrane. So remember, if we have straighter, denser, more compact components, we have a more rigid membrane. If we have a less dense, less fluid, less stackable components, we have a more fluid membrane. Before we move further and talk about how our cells regulate their density of the membrane, I do want to say that even if the membrane is in its densest state, even if a membrane is as rigid as it can be, it is not static. Still, the lipids are moving around constantly, proteins are moving around constantly. We're really just talking about the level of rigidity. The average cellular membrane has the viscosity of olive oil, and so a little bit denser than olive oil is still not very dense, and never should you have the idea that even a rigid membrane is static. It's just that the more fluid the membrane is, the more things are moving around, that's all. So how do our cells increase the rigidity of their membrane? Well, we do it largely by cholesterol, intentionally so. Cholesterol can increase the order and rigidity of a eukaryotic cell membrane, and it's pretty much done in this way. As you can see, because of the bulkiness of the head groups, the hydrophilic head groups, even saturated fatty acid tails, even phosphoglycerides that contain saturated fatty acid tails, can't get really close together because of these bulky charged head groups. However, cholesterol is very planar. It really is more of a two-dimensional than a three-dimensional molecule, and that planarness allows cholesterol to slip in between the fatty acid tails, as we see here. And the head group, the hydrophilic group of cholesterol, is nothing more than a lowly hydroxyl group, which takes up very little room. So cholesterol slips in between the fatty acid tails and makes van der Waals interactions across those fatty acid tails, while adding very little to the hydrophilic head groups and taking up very little room there. So cholesterol stiffens up cell membranes by essentially slipping in between fatty acids like a playing card and interacting favorably with the fatty acids on either side of itself through van der Waals interactions, thereby stiffening up that cell membrane. Heat can also play a role. The hotter an environment is, the more fluid its membrane will become. The colder of an environment, the less fluid the membrane will become. As soon as a lipid bilayer begins to quote-unquote melt, that becomes a positively cooperative process meaning it becomes easier and easier for it to continue to disrupt. So as we go from a low heat, more gel-like consistency, if we heat that membrane, we get much more liquid, and we do so more and more easily as the membrane loosens up. We already talked about the curvature of the lipid bilayer. We have larger head groups on the outer layer and smaller head groups on the inner layer, allowing that curvature to form naturally rather than it have to be induced. So now with a sense of what the lipids are doing, both the phosphoacylglycerides as well as cholesterol, it's time to introduce membrane proteins in general. In general, there are two classes of membrane proteins, and the classification depends upon how those proteins interact with the membrane itself. We have some proteins that really just transiently interact with the surface of the membrane. They're doing nothing more than making hydrophilic or charged contacts with the hydrophilic head groups of the membrane lipids. We call those proteins peripheral proteins because truly they're just on the periphery of the membrane. The other class of proteins literally embed themselves within the lipid bilayer itself. They are anchored to the membrane through hydrophobic contacts with the hydrophobic tails of the lipids of the membrane themselves. Because these proteins actually integrate themselves or enter the membrane, they're called integral proteins. So here we have some examples of each. Number 3, of course, this green protein is a peripheral protein. You can see it's interacting with nothing more than the hydrophilic head groups of the membrane. And peripheral proteins can either interact with the inner leaf or outer leaf of the membrane. They can be on either side of the membrane. Proteins 1, 2, and 4 are all integral proteins of different kinds. Protein number 2 exists entirely within the membrane. Protein number 4 has a domain which reaches beyond the outer leaf of the membrane, but it is integral. And protein number 1 is a very unique class of integral proteins that we will return to time and time again in this course. This is referred to as a transmembrane protein, and it's transmembrane because it penetrates and spans the entirety of the membrane. We have a region of the protein that exists on the outer face of the membrane, a large intermediate region that makes hydrophobic contacts with the tails throughout the entire interior of the membrane, and another domain of this protein that exists outside the membrane as well. So this spans the entire membrane, and we call it a transmembrane protein. Most of our signaling proteins are going to be transmembrane proteins, where the domain outside the cell hears the signal molecularly, and the domain inside the cell transmits that signal to the cellular interior. Again, most of these peripheral proteins are making contacts with the head groups through charge, because those head groups of the membrane lipids are charged themselves. Those can either be full electrostatic interactions or hydrogen bonds, or both. The integral proteins are relying heavily on hydrophobic forces to stay where they are. They are making hydrophobic contacts with the hydrophobic tails of those membrane lipids. Most of these transmembrane proteins, these integral proteins, have largely hydrophobic alpha helices made up of largely hydrophobic amino acids, and those helices are what's making these contacts with the hydrophobic tails. So if you imagine that most of the amino acids within this alpha helix were hydrophobic and uncharged, then every time a side chain projected out towards these hydrophobic tails, there would be a favorable hydrophobic or van der Waals interaction there. Because of that, this kind of keeps these proteins anchored within the membrane and stops them from floating up or down relative to the membrane. So we've talked about, for most proteins as they fold up, we see that they have their hydrophobic amino acids in the interior of the protein and the hydrophilic amino acids on the exterior. This is a bit counter to that because here we see hydrophobic amino acids on the exterior of the protein, the exterior face of the membrane-bound protein, and those amino acids need to be there to make these contacts with the hydrophobic tails. So what do membranes do? Well, as we said, they do a lot. They are the gatekeepers of the cell. They're responsible for receiving information from outside the cell and transmitting it inside the cell. And almost all of that work of the membrane, what membranes actually functionally do, is handled by membrane proteins. The membrane lipids are truly just serving the boundary or barricade role of the membrane, whereas it's the proteins that do all of the specialized work. It's transport proteins that serve as the gatekeepers. Receptor proteins are the class of proteins that transmit signals. Receptor proteins can interact with hormones, among many other things. They can be responsible for launching an immune response. They can be having metabolic information transmitted to them, where the information is whether glucose should be released from storage or absorbed from a recent meal. Even the fight or flight response is regulated by receptor proteins, having our bodies respond to potential danger. We will see as we go through this course that we also have membrane-bound enzymes responsible for metabolism. In fact, much of the final stages of glucose metabolism happen right at the mitochondrial membrane, and that is because most of those proteins responsible for those late stages of metabolism are bound to the mitochondrial membrane. So just as the lipid composition of the inner and outer faces or leaves of the membrane were different, so too is the protein composition. We see different proteins on the different faces, especially for the peripheral proteins and the non-transmembrane integral proteins. So different sets of proteins lie inside and outside the layer, and those different proteins give different functions to the inside and outside layer of the membrane. We call this general principle asymmetry. Our membranes are asymmetrical. The inside face is different in the size of the head groups, as well as the composition of the proteins, as compared to the outer face of the membrane and the size of their head groups of those lipids and the composition of the proteins we find out there. So this leads us now to the fluid mosaic model. As we've already said, even for the most rigid membranes, membranes are not completely static, completely rigid structures. They are dynamic and constantly in motion. In fact, lipids and membrane proteins are constantly diffusing laterally, and that means moving side to side and front to back. As I've already told you, the viscosity or the density of the standard eukaryotic membrane is about that of olive oil, and so there's a great deal of fluid movement. Now, olive oil is not very firm, obviously, but it's also important to note that it's 100 times more viscous than water. The fluid mosaic model essentially tells us that membranes are two-dimensional. Remember, not three-dimensional because of the asymmetry. We can't have molecules moving up or down. We can't have molecules flipping upside down, but we can have molecules moving front, back, left, and right. That's that lateral diffusion, and that molecules do move that way constantly in a very fluid way. So this model is meant to suggest that you think of a mosaic, but now that mosaic, imagine it in motion. Imagine these individual colored tiles were free to move or diffuse forwards, backwards, left, or right. What these tiles cannot do is float up and out of the mosaic, fall deeper into the mosaic, or turn upside down. They can't do that. But they can move forward, backward, left, or right. And that's exactly what these proteins can do as well. These proteins can float forwards, backwards, left, and right, like an iceberg in the ocean. So again, as I've mentioned, all of this diffusion is lateral. What is not possible in a membrane is transverse diffusion. Transverse diffusion is the toppling, or the reversing, the upside downing of these membrane lipids or membrane proteins. Again, if we think of the membrane as an ocean with icebergs, the proteins are the icebergs and the water are the lipids, we can have icebergs float forward, we can have them float back, they can float left and right, but what icebergs do not do is flip upside down in the water. And so we can have rapid lateral diffusion in a membrane, but we don't have this flipping of constituent parts upside down. That's true for both the proteins and the lipids. While these lipids are moving in matters of seconds, the transverse diffusion, the flip-flopping of these components, occurs about every several hours. And so, because membrane lipids as well as membrane proteins are pretty much locked and conserved onto their own faces of the membrane, not only do we see asymmetry of the membrane, we preserve that asymmetry. The outer and inner leaves are different, and they remain different because their two parts never intertwine. You've probably heard of cell membranes already referred to as semi-permeable cell membranes. And by semi-permeable, we mean that some things can permeate the membrane, some things can float right through, but other things cannot. In general, molecules that can float passively through the membrane like a ghost through a wall are very, very small molecules that don't have full charges, and pretty much any molecule of any size that is largely hydrophobic. Those things can pass through the membrane without any aid. Most of the molecules we encounter in life, though, are either charged, partially or fully, or are very, very large, and those molecules cannot pass passively through the membrane. Instead, they require a doorway or a channel to do so. So as we move on now to transport across the membrane, we have two classes that we need to keep in mind. Both of them require a doorway, both of them require a protein that allows molecules to flow through, but sometimes those molecules are following their concentration gradient, and sometimes they're moving against it. When a molecule requires a protein in order to enter or leave a cell, because it is either too charged or too large to do so on its own, but when that molecule moves, it is following its concentration gradient, moving from high concentration to low. We call that passive transport. It is transport because it required a protein doorway, but it's passive because it requires no energy. It's following its own concentration gradient. When a molecule cannot passively diffuse across the membrane on its own and requires a doorway in order to cross the membrane, and on top of that, it's moving against its concentration gradient from low concentration to high, that is active transport. Transport because it required the doorway in the first place, and active because it is an energy-consuming process as we pump things against their concentration gradient. So if these white molecules could not float across the membrane and required a channel or pump to do so, if they required a pump or a protein to get across the membrane, if the molecule was moving from left to right, we would be dealing with passive transport. If the molecule was moving from right to left, that transport would be active. Active transport requires energy because most molecules want to move away from a crowd. If you're trying to force molecules towards a more concentrated environment, you have to force them, which requires energy. So to review it, passive diffusion, again, not transport, passive diffusion is a molecule simply crossing the membrane all on its own. Oxygen can passively diffuse into our cells. Oxygen is small enough and uncharged enough that it requires no protein doorway, it requires no channel or pump, it simply moves from high concentration to low concentration, and it moves through the membrane as though the membrane wasn't there at all. Very few molecules can do this, but oxygen and water are some of the ones that can. Then we have facilitated diffusion. Facilitated diffusion occurs when the molecule requires a protein doorway, but that doorway only allows the molecule to move with its concentration gradient, from high concentration to low. This diffusion is passive in the sense that it doesn't require energy, but it needs to be facilitated. It needs to be helped along by the presence of this doorway. We call proteins such as this channels because they're doing no work, they're simply allowing molecules to move the way that they would be moving anyway. An example of this is glucose. Glucose is too large and contains too much partial charges to simply float through the cell membrane on its own. It requires a protein channel to do so, but glucose largely moves with its concentration gradient, and so it doesn't require energy. And then we have active transport. Active transport is when a molecule cannot diffuse across the membrane. In this case, a positively charged proton is a good example. Proton can't diffuse across the membrane because of that strong positive charge, but additionally, the cell is pumping that molecule across the membrane from low concentration to high. Well, if you're going to be pumping something, if you're going to be moving something against its concentration dependent, then that's real work, and so you need energy. So here it is the energy from ATP in this example that allows the cell, the membrane pump, to move these protons from low concentration to high concentration. Most of the time the energy for doing this comes from ATP, but there are times where the energy can be harvested in other ways. Secondary active transport leverages the energy created by one molecule following its concentration gradient to drive another molecule against its concentration gradient. We won't go too much into that here. We cover that a little bit more in cell and molecular biology. but do know that ATP is not the only source of energy for active transport. So to summarize what we talked about here, we began this lecture half talking about cholesterol. Cholesterol as a molecule is not only the precursor for a lot of our steroid hormones, it also allows eukaryotic cells to increase the rigidity and density of their membrane by slipping those planar cholesterol molecules in between fatty acids, creating more and more van der Waals interactions between those fatty acids, allowing that membrane to stiffen up. We moved on to different classes of membrane proteins in general. Peripheral proteins are proteins that do not penetrate the membrane at all. Instead, they act only with the hydrophilic head groups on either side of the membrane. You can have peripheral proteins inside the cell and peripheral proteins outside the cell. That leaves integral proteins as our other class. Integral proteins do penetrate the membrane itself, and what anchors them there are hydrophobic interactions that portions of the integral proteins make with the hydrophobic tails in that membrane. We have a special class of integral proteins, the transmembrane proteins, which completely span the membrane. They have exterior domains on either side of the membrane and then a large interior domain that's actually making those hydrophobic contacts. We talked about the asymmetry of the cell membrane. The set of proteins on the interface of the membrane is distinctly different from the set of proteins we find on the outside layer, and the same is true of the lipids themselves. We have different types of lipids on the inside and outside. Smaller, less bulky head groups inside on the inner face of the membrane, larger, bulkier head groups outside on the outer face of the membrane, and this contributes to the natural curvature or spherical shape that membranes take. We wrap the lecture up with some discussion on transport. We have pumps that move molecules against their concentration gradient across a membrane. We have channels that allow molecules to move across a membrane while they follow their concentration gradient. And then we also have the potential for passive diffusion, where molecules don't require a protein at all to transport across the membrane. They can passively diffuse all on their own, again, like a ghost through a wall. So that's all we have for lipids and biological membranes here. We'll be shifting gears again now, fully poised to begin the early stages of glucose metabolism. In our next lecture, we will begin exploring some of the fundamental concepts that we need to be comfortable with for metabolism, setting the stage to cover metabolism proper soon after. As always, thank you for watching.